Yes, I'm Nick Knobloch. I'm a scientist at BioMarin Pharmaceutical from the genomics team. Sorry, yeah. Is that good? Okay. And I'm going to be talking about some work that we've done to what we think conclusively demonstrate that haploinsufficiency underlies the neurodevelopmental consequences of SLC6A1 variants. Okay. Ooh. Way too fast. Okay. So yeah, just want to, before we get into it, I want to express that this was work that was done as a collaboration with BioMarin and with UCSF, led by Marina Trinidad, who is now a PhD student in Jennifer Doudna's lab, Stephen Froelich, Arthur Worcester, Jeff Bergwig, and others at BioMarin, and then by a really great team at UCSF, led by Stefan Sanders. I also want to thank some folks from the University of Southern Denmark and the Econ School of Medicine, but maybe I think most importantly, the SLC6A1 patients and the variant depositories, that made all of this research possible. So, mutations in SLC6A1, were originally associated with epilepsy dating back to this Carville paper in 2015. And a few years after that, folks working in the Autism Sequencing Consortium found that there was this great enrichment of missense variants in excess of protein truncating variants in SLC6A1, which was somewhat unusual for neurodevelopmental genes. And then a couple of years later, some folks identified that, in fact, among neurodevelopmental delay genes, that there's some mild evidence of enrichment for variants associated with schizophrenia. So, I think as we've heard about a lot today, there's like a really wide-ranging number of phenotypes associated with SLC6A1. And so one thing that we wanted to kind of get at with our study was, for many genes like SLC6A1, when you observe an enrichment for missense mutations, one often associates that with a gain-of-function mechanism. And originally, when in the Carville 2015 paper, it was they hypothesized that the missense mutations lead to loss of function and reduced GABA uptake, but we wanted to really comprehensively test a very large number of variants and figure out what experiments would most conclusively show that haploinsufficiency is really, how universal is that mechanism? Are there gain-of-function variants that perhaps we're not seeing? And then if we're not seeing them, is it because they don't exist, or perhaps there's some sort of some sort of prenatal lethality, so kind of what's going on where we're seeing, in many ways, haploinsufficiency via missense variants and haploinsufficiency via truncating variants, should have sort of a similar mechanism. And so why are we seeing, this enrichment for missense variants? And then, the last piece of it is, there are a number of folks, because of the de novo nature of the disorder, they have variants of unknown significance, right? And so we wanted to, molecularly test these variants so that we can see, are these more like some benign variants that we know exist, or, okay, we're still having problems with them. Yeah, don't be afraid of the mic. Okay, yeah, or are they more like pathogenic variants? So what did we do? So we took 213 SLC6A1 variants from a number of different patient sources, and these were cloned into a plasmid with what's known as a BLA reporter, and these were created via site-specific mutagenesis. We had this automated nucleofection pipeline into a monoclonal HEC cell line that is SLC6A1 deficient. And for a subset of those variants, we were able to get subcellular localization via immunofluorescence. And so that would tell us kind of like where in the cell, or if in the cell, SLC6A1 is being expressed. And then we, am I still?
Slightly a little bit louder.
Okay, yeah, sorry. And so we followed the transfection by incubation in a deuterated GABA to measure GABA uptake. And then all of that was followed by LCM-SMS to actually quantify both the GABA uptake and the BLA activity, which will give us like the level of SLC6A1 expression. So there were 24 control variants that we used, consisting of 6 synonymous and a wild type. There were these 10 missense variants from a population database that are thought to be controls. Like, we don't really know, the kind of complete phenotype in these individuals, but they are not considered pathogenic variants. And then another 7 variants that were predicted to be benign missense from ClinVar. And so we used the sort of GABA levels of these 24 variants to kind of set a control, right, for a reference range for GABA uptake. And then we found that with the 213 missense variants that we tested, 100 of these showed severe loss of function. So that's like a decrease of 82% or more compared to our reference set. Another 25 showed an intermediate level of loss of function. 85 were in the typical range, and then we only found 3 that showed an increase of 47% or more from our assay. And we took 68 of those variants, we tested them in-house at BioMarin and at UCSF, and they showed a pretty good concordance. So we're feeling pretty good about that. Taking a look at where these loss of function variants distribute across SLC6A1, you can really kind of see, you can see they're kind of all over the place. Except maybe that like some of these typical activity variants look like they're a bit more towards the outside of the protein versus the inside, where we're seeing a bit more enrichment of the severe loss of function variants. But when we overlay that GABA uptake data with the surface expression data, we found that we really find that there are 2 broad categories of loss of function mutation. So there are a set of variants where there is no GABA uptake basically because there is no protein expressed at the cell surface. And then there is another set of variants that lead to a protein that is trafficked to the membrane, but then is not working. So I kind of think about it as like, if you think about the protein as maybe like an elevator, you're installing the elevator, like, maybe the elevator can't fit in the elevator shaft, and then you're obviously, not gonna be moving anything, or maybe the elevator fits, but then the doors don't open, right? And so we think that like, the SLC6A1 structure is really sort of determining the variant function, and that variants on the interior of the 3D structure that are like the closest to the interior are most likely to disrupt function, while those that are on the surface often have a milder impact. And then among those that are kind of the variants that are deep within the protein core are usually reducing GABA uptake due to poor trafficking or by sort of altered interactions with the transporter complex, whereas those that are more like directly involved in binding of GABA, many of those are able to traffic to the cell membrane and then aren't functional when they get there. So we found that of the 100 so variants of unknown significance that we tested, about half of those had protein levels that were similar to those we found in these like known pathogenic variants, or had dramatically reduced GABA uptake relative to our controls. We also found, consistent with what you would expect, the known pathogenic variants had the most severe average decrease in GABA uptake relative to the controls. And then as you kind of lose confidence at the clinical level as far as variant annotation, that was like concordant with what we found with the assay. Yeah, so getting into these 3 variants that showed a gain of function, right? So like an increase relative to wild type in the GABA uptake of more than 47%. So one of them was a control variant from Nomad. And I say control, like, should sort of be control in quotes because, Nomad is this population sequencing database, and, we know that they were control for some sort of genetic study, but we don't know, really the phenotype of that individual, so we can't really say, for sure whether or not we can really think of it as a control variant. Another one that was also present in Nomad and has been associated with an individual with early onset epileptic encephalopathy. We didn't get a chance to retest that. And then another variant of unknown significance. And so we took the top 10 cell line, the top 10 variants with the highest sort of GABA uptake, and we retested those by establishing stable cell lines. And we found that uniformly there was lower uptake. So when we kind of repeated this experiment in a higher quality, using a higher quality system, this gain of function is basically eliminated. And so there's kind of a non-significant trend overall as being above wild type levels. So now that we had like hundreds of variants that we had tested the GABA uptake of, we could then expand outside and train a linear model for any possible missense variant in SLC6A1. And we found that simply using the ClinPred predictor gave us like quite robust performance in predicting the GABA uptake just from the ClinPred score alone. And so to kind of think about why we're seeing this missense enrichment in SLC6A1, we generated these ClinPred predictions for every missense variant in SLC6A1, and then for like several other comparable genes associated with neurodevelopmental disorders. And we find that like SLC6A1 is basically like the most fragile to missense prediction, to missense variation. And that this is like a highly significant. And so it really seems to be that missense variation is just much more likely to be damaging in SLC6A1 as compared to other genes. Oops. Yeah, and so then another maybe piece of that is that we found that this GAT1 vulnerability, right, like to missense variation, it explains this enrichment of rad missense variants, but it doesn't fully account for these like recurrent de novo mutations. And we found that when we looked at the mutation rate of these variants, we found a correlation between the estimated missense variant mutability and the predicted GABA uptake. And then thinking about whether or not like CPG sites maybe were kind of a cause of this, only like 2% of those of variants are at these hypermutable CPG sites, but 71% of all of the recurrent variants and all variants in 3 or more individuals are coming from hypermutable sites. And so even though there are a large number of different variants, right, for the condition, they concentrate in a smaller number of patients. Yeah, and so we're able to predict GABA uptake pretty consistently using this ClinPred score. And we also find that, these hypermutable missense variants, they are uniformly predicted to result in loss of function. So yeah, just to kind of summarize what we did here. So we assayed 213 unique variants, and we found that essentially all of them lead to reduced GABA uptake. We found that the haploinsufficiency of SLC6A1 is mediated both by lower surface localization and by lower uptake, and that this missense enrichment paradox is kind of coming from the high sensitivity of GAT1 to missense variation, and that the recurrent de novo missense variants are driven by both loss of function effects and by these hypermutable CPG sites. So yeah, I think, as far as like clinical implications, I think we've shown that like this is a relatively scalable system that is like, I think it provides strong evidence for reclassifying some of these variants of unknown significance as perhaps being pathogenic. So yeah, this is all in a American Journal of Human Genetics paper. So if folks would like to read more, they can scan the QR code. Thanks.